Which paper are you starting with?
Let's take a look at which paper we're starting with. It's the Proteomics papers. The first paper is by Shimada, root R, the root R transcription factor. So there's 4 papers here. Root R transcription factor binds targets within genes. Martinez writes a paper, characterization of the Streptococcus cerebrinus acid stress response by interspecies microarrays. And Balagan, growth phase dependent modulation of RGG. Rochat talks about genome-wide identification of genes regulated by SPX in subtilis. Okay, so the first thing, let's take a look at something funny. Here's some molecules you might like to look at. The first one is orthodox. So in a benzene ring, if 2 groups are next to one another, they're in ortho position. So orthodox. See, there's 2 PhDs on orthodox. Paradox is they're in opposite sides of the benzene ring. There's also the Mercedes benzene. If you have neons all in a circle of 6, it's a cyclone. The anticyclone is neons backwards. Dimethyl chicken wire. Now, if things are in meta position, you can have the number 4s in a meta position. See, it's so meta, meta 4. This here spells stasis, S-T-A-S-I-S. They're in metastasis. So those are some interesting molecules, right? Just something silly to start off the presentation with. Okay, so I said I'd have 4 papers for you. The first paper is the root R transcription factor. Now, root R, what is it? It's called root R because it's pyrimidine utilization. They're taking the R from pyrimidine and UT for utilization, and the letter R is because it's a repressor. So it's the root repressor. Now, root repressors bind transcriptional, in a transcriptional manner. They do it in a dual fashion. That is, there's 2 molecules there. You can't really get this from the picture, but there's one on top. If I had colored it differently, there would be one on the bottom. These molecules also bind uracil, and you can see there's a uracil bound inside of here, inside of the cleft. These molecules, the root R molecules, regulate the degradation of purines, and they're also involved in pyrimidine metabolism. They belong to the TET-R family, and they contain this transcription factor motif, the helix turn helix motif, and a C terminal for dimerization. That's the overall structure of root R. In this paper, I'm looking at an experiment called chip-chip experiments. It's for chromatin immunoprecipitation on a chip. That is, if you take a look here, you've got your genomic DNA, and the genomic DNA attaches to a protein of interest. What you do is you break up the bacteria, cross-link it, and shear it. You're left with the protein of interest bound to DNA, and then you immunoprecipitate it. So if you have an antibody, you bind that to your protein of interest. The protein of interest then is degraded. All you're left with is the DNA. You fluorescently tag the DNA and attach it to a microarray, and then what you do is you get the microarray expression data. So this shows you which parts of the DNA are attached to your proteins. That's a chip-chip experiment. You can also do chip-seq experiments, where you do the same sort of process and then sequence the data as opposed to putting it on a microarray. You could also use this for ribosomes and do ribosome binding sort of things. I took this picture from Wikipedia because it's an easy picture. Okay, so here's the E. coli chromosome. You've got about 45 megabases. And what they found was root R binds across the chromosome in a multitude of positions, and you can see very specific positions are bound. Now this one's
this is from their chip-chip.
right. This is the chip-chip experiment, showing the binding positions along the chromosome. Now, when you look at the regions that they're bound, letter B here shows that they're bound to intergenic sequences. So between DAP-B and CAR-A, you can see it's bound in between those two genes.
Mm-hmm.
That's in the presence and absence of uracil. Root R, root A, presence and absence of uracil, and so on. In letter B, in letter C, you can see that root R also binds not just to the intergenic regions, but also to the coding regions. So it binds within genes. Now, in the next figure on the paper, what they show is the root R binding sites are identified by chip-chip analysis. And what you can do is then analyze the binding sequences and figure out what the consensus is for the binding sites and actually match it. And as you can see, sometimes the scores are pretty good. It's like 13 out of 14 match, 11 out of 14, but they go all the way down to no consensus site, but it still binds, or 7 out of 14, and it still binds. So sometimes there's strong binding, sometimes there's weak binding.
And this is also still from the chip-chip?
Yes, this is based on the chip-chip experiment. And you can also see that they've categorized these reactions: DNA, RNA related, metabolism, function unknown, drug resistance, transport. Here's an EMSA experiment. So in this figure, we've got in letter A, high affinity targets, and letter B, low affinity targets. Now, in these genes, you can see as you add more root R, the binding should make more like a stronger complex.
Mm-hmm.
Down at the bottom in letter B, although these are found to form binding complexes, the formation of the complex is very weak. So there's 4 genes with weak binding sites and 16 with strong binding, high affinity sites. And in this figure, what they're showing is that you've got this root R transcription factor. It's found within a lot of gram-negative bacteria: E. coli, Salmonella, Shigella, Citrobacter, and so on. Isn't Enterobacter a gram-positive? Klebsiella, gram-positive? I think so. Testing my knowledge as I go by. Anyway, so the root R is in very many bacteria. Now, how is it distributed for its binding sites? What they did was they took a look at several genes, LEX-A, FUR, FRUR, CRP, FIS, and said that, okay, where do these genes bind? Well, they bind within the intergenic regions and not so much the coding sites. Root R, 90% of root R actually binds on coding regions. And they often bind away from the start codon. So it's kind of weird. This has not been seen before.
Binds the start codon, away from the start codon?
Binds away from the start codon oftentimes, and binds within the gene. So it's not just...
Is there significance to binding intergenically?
Well, if it's the intergenic region, that it can prevent or stop transcription of the gene.
So this is binding downstream of the +1 start site?
Inside of the gene, yeah. Mostly. 90% of the time it does.
That's interesting.
Yeah. They don't know why that is.
Wait, 90% is in coding. So
In the
intergenic.
coding, in the coding region. Not the intergenic region. That's 10% of the time it's in the intergenic region. 90% of the time it's within the gene.
Well, it's downstream of the +1
Yeah.
start site. So the translational start site. Okay. Binds. So what do you think it could be doing when it binds downstream?
Well, if it's binding
Or
downstream,
in.
it's preventing the translation of the gene in some manner. So if it's pyrimidine metabolism, obviously it's going to detect how much pyrimidines are in the system, and if there aren't enough, then it'll allow the translation of these things to go. Probably.
So, Navarre, so I guess when he was a postdoc or a PhD, he worked on, you know how he works on the histone-like proteins? So, I forget the transcriptional regulator, but there's a transcriptional regulator in Salmonella that does bind downstream of the +1 start site, and he's shown that it kicks off the basically remodels the DNA structure to
Right.
allow transcription to go through.
So maybe the opposite? Or it's no.
I guess that's when
inhibits.
it's activated, that could inhibit.
Timing is really
Oh, but
important
you-
for these things, as well as location. Yeah.
Yeah. It's an inhibitor in most cases.
Repressor.
Right?
Yeah.
So it probably repressor. So it probably just represses translation. Right? That's fine.
So that's that paper by Shimada, and the next paper,
Oh,
Martinez.
really?
Oh, that's what you meant 10 minutes. You just have to present that.
Characterization of Streptococcus sobrinus, acid stress response. And this is a paper
Is
that
there a related protein like that in strep mutants?
root R.
Yeah. Can you? I guess.
I didn't look.
I guess
Pyrimidine
you have to
metabolism
do
is done
like-
by the pyre genes, so I don't know if there's a repressor to the pyre genes or not.
I wonder if there's a set of proteins that do this, like or
I'm sure we
regulators
could.
that bind inside.
The chip experiment or chip-seq experiment could be done for any protein transcriptional factor that you want to look at. Yeah.
Were you saying something about intergenic regions, like short non-coding RNA sequences?
Yeah.
So could it not be transcribing intergenically something? Like, what is the significance of the intergenic binding? So, okay, there's binding in, like, the coding sequence of the genome, which obviously prevents translation. Then it regulates transcription.
I have a great picture. I'll show you after I'm done. Okay.
Oh, okay.
I know it's kind of a cop-out, but actually, you asked. Why don't I just show you?
Yeah. You like
Wait, are we
wanna
supposed to
stick
ask
to
the question?
Yeah, I think so.
Or
no?
are we supposed to
Don't
save
you
it
want
to,
to discuss?
or is it just supposed to be a presentation?
This is for next time.
Oh, you're gonna have to present this.
No.
I will.
This is him presenting it to us, right?
So what's the point of it binding on the intergenic region? So let's say you have a protein like MAR, R, it dimerizes and binds to intergenic regions that have this inverted repeat,
Mm-hmm.
and it prevents transcription of adjacent proteins. So the thought is that transcription factors can prevent or promote transcription of the genes of interest.
Through regulating short non-coding RNAs? Or
Well,
So
In this
Or
case,
in
they're
this
just
case, it's an
regulating-
It's
intergenic
just
regulatory region.
So they're just regulating the promoters
Okay.
and who binds there. So yeah, they're sitting there and blocking RNA polymerase from binding. But yeah, there is a lot of
like research
short
on-
no coding RNA that gets transcribed, or even within genes, like the
I
COMEX.
found with my microRNAs when I was studying in intergenic region, the microRNA gets co-transcribed with the gene and it regulates the gene too.
Yeah,
And it negatively inhibits.
Yeah.
yeah.
That's what we, like when I was doing research, so I was just curious to see if it was in bacteria.
Oh,
Oh.
lots of it. It's huge. And sometimes I think even Andrew is finding that there are different transcript lengths that get transcribed. So it'll be like one long one shorter one, and they often have different functions as well.
There's a paper out for S mutans. They found about 700 microRNAs in strep mutans.
Even when we did,
So
we did
20 to 25 nucleotides long.
Mm-hmm.
They're- There's all of this stuff there. They don't know what it does. There's no function, there's nothing known about them in mutans yet.
That's pretty cool.
Even
But
when they
in
did-
humans,
You guys
they're
are
really
doing
finding really cool stuff. We're
Yeah.
not doing it, but other people are. So here we go, Martinez, the characterization of Streptococcus sobrinus. What is sobrinus? Apparently, it's a primary organism responsible for caries. It's found along with mutans to cause cavities.
Yeah.
Now it's found on smooth surfaces. It has an ability to tolerate acid as well as just very similar manner that mutans can tolerate acid. However, it's really tough to work with in a genetic way, so that's why it's not very well commonly used. Here's a 2D protein at pH 7 and pH 5. So what they've done is they've looked at the proteins at pH 7 and tried to figure out which proteins are differentially expressed. And they've got these two, SS1, SS4, and if they're underlined, they don't really know confidently that that's actually different than pH 5. I know it looks different here, however, they're in the paper, they don't confidently figure these out. At pH 5, however, there's a number of proteins that they've confidently identified. In the next table, I show a bunch of proteins that they've confidently identified as being involved in the acid stress response. Now, if you take a look at this, it's a lot of glycolytic genes and also malolactic acid fermentation genes.
Mm-hmm.
In the next figure, what they do is they do malolactic fermentation of Sobrinus and mutans and compare the two. And then, sure enough, they're very similar in the response. The acid tolerance response is very similar. And that's that paper.
Wait, what's malolactic fermentation? I don't know.
Yeah.
What's malolactic? What is
Malate,
malate?
right, and they're fermenting that sugar.
What's malate?
What is malate?
It's probably a really stupid question. But I don't know. Do you know?
Malolactic fermentation is a process in winemaking which tart-tasting malic acid, naturally present in grape must, is converted to lactic acid. So
Oh,
they're taking malate and converting it into lactic acid.
Oh.
Okay.
So how do they do this? They provided. So they expose the strep to malic acid.
It's right, just like our acid tolerance assay.
Yeah, but instead of like lactic acid, they use.
It's a different sugar that they're starting off with. Instead of us, we always use glucose. They're using a different starting carbon
Do they explain why?
to show that it's very similar to mutans.
Yeah, but why not use a different? Like, why not use the standard acid tolerance? You know?
I'm not 100% sure at the moment.
Okay, yeah.
Thanks.
No, I just don't know. They probably.
They're trying to make a comparison between mutans and characterize it closely with Sobrinus.
What is this big table that they have here? Genes.
Those are the
sequencing.
genes that are
Why are they? What's in? They're looking at expression using interspecies microarrays. strep mutans genes. No?
Yep.
So they're looking.
We're looking at the paper. What was the author's name?
Martinez.
I'll look at the table as well. Let's see. Interspecies microarrays. Not only different species microarray, but also pH5, pH7. I thought this whole table was pH5, pH7. I wasn't thinking that it's interspecies, but that's what they're saying, interspecies.
I just. I guess I should have read this.
And
Because I guess I'm not clear of what an interspecies microarray is. So you expose, because they're looking at Streptococcus subrinus here, but all the genes that are upregulated are mutants. So are they taking subrinus and
adding
whatever
it,
is upregulating and then testing it on a microarray of strep mutants? So are they identifying genes that are homologous, that can bind to a strep mutants? You know what I'm saying?
Yeah.
I don't know what an interspecies microarray is. But I guess that's what it seems like to me. Yeah, they did. Microarray slide, strep mutants, to determine whether strep mutants UA159 microarray slides were suitable to study the transcriptome of subrinus. We hybridized subrinus genomic DNA to the strep mutants. So I think, yeah.
so they just used the mutans
Yeah.
chips and did a microarray with the mutans chip. I wasn't going to talk about the
So I guess that's what they
microarray.
had available. Okay.
This is done out of Rochester, correct?
I'm not sure.
I think it's the it's Jose Lemos
Yeah.
with Julia A. Branches.
Mm-hmm. Okay.
So they would have the microarray, mutans microarray, available to them, and they just tested the sovereignness
So it's
against
more
them.
of like a technique type paper. So you can
Yeah.
look at the microarray of subrinus by looking at the using
The mutans microarray. Yeah.
Got it. Cool.
Okay, next paper. And Balagan talks about RGG binding specificity in pyogenes. Okay, so what is strep? Pyogenes, well, it's a group A strep infection causing bacteria. It displays group A antigen on its cell wall. And what is RGG? Another name is ROPB. It's a transcriptional regulator. It's another transcriptional regulator. It has a helix-turn-helix DNA binding motif in the amino-terminus, and it includes what's referred to as a tetratricopeptide repeat. And I just gave a Wikipedia example of what that is there. One of the reasons I thought this would be interesting is because there's a novel double tryptophan peptide pheromone controlling competence in Streptococcus with an RGG regulator. This is something that we in our lab are looking at because this RGG regulator is involved with COMX and COMS, COMR, XIP regulation. Okay, so in the first table in this paper, what we look at is chip fold enrichment, and they identify the genes that are associated with RGG. So these are non-coding regions that bind RGG. Again, I showed that picture of a chip-chip experiment, and here what they're doing is just finding the regions along the chromosome that are attached to RGG. Now, what they're saying is, in the next figure, with the chip-chip experiment, they can say that the non-coding sites are, there's more of those than there are coding sites, but it does bind coding regions and it does bind non-coding regions as well. So they found RGG bound to 62 places on the chromosome. Then they kind of looked at which genes were associated, and they're all over the place: repair, transport, metabolism. There's some non-coding, some coding, like you saw in the last picture.
Right.
What they then do is they perform a couple of EMSAs with those promoters, and you can see that with very little, let's say PEMM49, you're not forming a complex, but as you increase the amount of this promoter region, you get a complex being formed. And they're using 0.1 to 2 nanograms, which is a very small amount. And PSOF, they find this complex. PSFBX, they're finding a complex as well. PSBEH, they expect a complex because it says it's bound, but they're not finding the complex being formed. Perhaps what they're not doing is not adding enough either DNA or maybe not enough protein. And the protein amounts are in picomole amounts of the purified RGG protein. And on the bottom are the controls with the cold unlabeled GROEL and cold specific competing DNA. Now, because of that experiment, they wondered, does RGG act in a similar manner as LAC-D? And so what they did was they performed a bunch of luciferase assays, and they found that the promoter is responsible for repression. And as you can see, RGG and LAC-D both operate in a similar manner. So if you knock out RGG and LAC-D, you get the same sort of profile, but if you complement it with the LAC-D, you get the repression back. So LAC-D and RGG operate in a very similar manner. That's the take home from this figure. Also, they figured that RGG and LAC-D work at the same time. That's in exponential phase, and that's shown in this next figure. So during exponential phase, when the bacteria is undergoing glycolytic flux, when it needs the sugar going into the bacteria for growth, what happens is RGG, which is shown in gray, and LAC-D1, which is shown in black here, they dimerize, and they also attach to the surface of Streptococcus pyogenes. And then, after the glycolytic amount is lowered, there's a conformational change with LAC-D1, and LAC-D1 unbinds from RGG, which then has different proteins bound to the surface.
You mean like D1 binds different promoters?
Binds different promoters at the surface, right?
Not at the surface, just...
Well, if RGG is at the surface of the bacteria...
This is the genome. RGG is a transcriptional regulator.
Yeah, I was thinking this is a picture of the cell, but you're right.
No, yeah.
But it would bind at the...
No, they just bind the promoters. So if there's RGG and LAC-D, and there's both of them in high glycolytic flux, then they dimerize, or they bind the same sites as a dimer. But when RGG is not there, the LAC-D will bind to other
other
promoters
locations.
and regulate something else.
Yeah.
I guess related to. That's cool.
And last paper. So I'll look across the whole chromosome of Bacillus subtilis and look at SPX. Okay, in this case I've drawn a picture with SPX. It dimerizes again. I've colored the two regions differently. What is Bacillus subtilis? It's a soil-dwelling organism and forms spores. It's very popular because it's like the E. coli of the gram-positive world. Lots of genetics is done on it. If you go to the subtilis wiki page, you can see 100 different conditions for the expression of your very individual genes. So let's say we're looking at SPXA. You can follow the expression of SPXA under 100 different conditions to figure out if it's upregulated or downregulated at different times. Also, what they've done are a number of transcriptional studies showing how the region is reacting to different types of stressors. So they've done the expression study as well as all of this gene expression study. So lots is known of each individual gene of Bacillus subtilis. And if you look along the chromosome, SMU 1142C is also named SPX. So is that a homolog? I'd have to do the alignment. I haven't done that yet.
There's Strep mutans has SPXA and then it has SPXB.
A?
There's someone works on these things. They're like competence related. I know because I looked at them before. Mutants. Chi-Fash. This is the Wagner Doppler, I think. No?
I'm looking for SPX 1142. 1142.
There's two of them, I think. Yes, SPXA and SPXB specifically interact with RNA polymerase to regulate gene expression in Strep mutants. Lemos. This is Lemos one.
Lemos work. Oh, nice. So SPX is important in mutans as well.
Yeah.
They're looking at SPX in Bacillus subtilis, trying to figure it out,
Oxidative
and they're
stress.
also trying to figure it out in mutans. Okay, what I've got here is if you knock out SPX and you stress it with diamide, and this is what the structure of diamide looks like, it doesn't grow very well. If you overexpress, if you put back the promoter under the SPA gene, you can recover SPX activity and bring it back to wild type. Here's an expression profile. So SPX on the Bacillus chromosome is very close to the MEK-A protein, and it looks like it's co-transcribed with YJBC, whatever YJBC is. And I've got the growth curves here showing that the growth for the mutant is very close to the growth of the wild type under normal conditions. This next figure is really complicated. There's so much going on. I'll just talk a little bit about this region first. So in the second line here, you can see they say green 5'. That means that they've figured out a 5' region, if you follow along with the green points. 3' is they found yellow 3' site. So by doing the expression profiles, they've actually called new 3' untranslated regions. They've also, oh sorry, this is red, 3' untranslated regions. Yellow is the 3' extended transcript after partial termination. So if I look here, this is yellow, so this gene is probably extended to this point after it's partially terminated. Black is a new gene and promoter and terminator being called. They've also done the expression profiles for forward as well as reverse for wild type. RNA-seq. The bottom is RNA-seq. And this middle part is chip-chip profiles. So they've also done an identification of where the transcription factor SPX binds to the chromosome. So that they've done the chip-chip experiment as well as RNA-seq in this paper.
Nice.
Yeah, lots. COM-R would be a great, wait,
And
COM-X. COM-X would be the best one to do. Wouldn't it?
Right?
Yeah. Because it's so interesting.
Although the binding site, the promoter is identified for COM-X, right?
There's
sites
still
where-
stuff we don't know. You could find out much more information using one of these experiments, I bet. So in this paper, what they did was they looked at the consensus sequence and they tried and figured out what the letters are for the consensus regions. And they figured that the -35 and -10 regions are extended for SPX regulated genes. And in fact, they've even worked it down to the -44 and -43 nucleotides. So if this is the -35 site, they've gone way back to the -44, 43 to figure out. So
This is like the -35 regulatory region of a gene.
Of a gene, yeah. Of a gene regulated by SPX.
So they're extended. But usually, I guess, -35, Oh, okay.
So they've figured out the consensus sequence of these two regions.
I see. Okay.
And like I was saying, you can figure out where the -44 site is by this number here. It's close to -2, so G is -2. This will be number 44, that's the G, and C -2, there's the C. They figured out the consensus of those two nucleotides. All that work just to figure out the consensus of the -35, -10 region for SPX. It's a lot of work. So in this next figure, you've got the effects of SPX on transcription. What they did was they used DTT. Here's a figure of DTT. So DTT will interfere with your disulfide bridges. So this column here, this is the positive control with two newly identified promoters stimulated by SPX. B, newly identified promoter stimulated by the oxidized form. C are your negative controls, and D is a new promoter stimulated by both the oxidized and reduced form of SPX. So A, PTRXB was already known, so this is your positive control, and it's known to be oxidized, and it's also shown to be reduced as well in the presence of DTT. PYUSI, oxidized and reduced. PYUGP, oxidized and reduced. Same thing for the letter B, oxidized by 4 different promoters. C, like I said, these are your negative controls. And D, it's oxidized and reduced. So PYVGN is both under the SPX control. And that's it. That's all of those.